Archaea.
The Archaea are a domain or kingdom of single-celled microorganisms. These microbes are prokaryotes, meaning they have no cell nucleus or any other membrane-bound organelles in their cells. Archaea were initially classified as bacteria, receiving the name Archaebacteria, in Kingdom Monera, but this classification is outdated. Archaeal cells have unique properties separating them from the other two domains of life, bacteria and eukaryota. The Archaea are further divided into 4 recognized phyla. Classification is difficult, because the majority have not been studied in the laboratory and have only been detected by analysis of their nucleic acids and samples from their environment. Archaean bacteria are similar in size and shape, although a few Archaea have very strange shapes, such as the flat and square-shaped cells of Hyloquadratum wallspit. Despite this visual similarity to bacteria, Archaea possess genes and several metabolic pathways that are more closely related to those of eukaryotes, notably the enzymes involved in transcription and translation. Other aspects of archaeal biochemistry are unique, such as their reliance on ether lipids in their cell membranes. Archaea use more energy sources than eukaryotes, ranging from organic compounds such as sugars, to ammonia, metal ions or even hydrogen gas. Salt-tolerant Archaea, the Halochaea, use sunlight as an energy source, and other species of Archaea fix carbon. However, unlike plants and cyanobacteria, no species of Archaea does both. Archaea reproduce asexually by binary fission, fragmentation, or budding. Unlike bacteria and eukaryotes, no species forms spores. Archaea were initially viewed as extremophiles living in harsh environments, such as hot springs and salt lakes, but they have since been found in a broad range of habitats, including soils, oceans, marshlands and the human colon and navel. Archaea are particularly numerous in the oceans, and the Archaean plankton may be one of the most abundant groups of organisms on the planet. Archaea are a major part of Earth's life and may play roles in both the carbon cycle and the nitrogen cycle. No clear examples of archaeal pathogens or parasites are known, but they are often mutualists or commensals. One example is the methangens that inhabit the human gut and the ruminant gut, where their vast numbers aid digestion. Methangens are used in biogas production and sewage treatment, and enzymes from extremophile Archaea that can endure high temperatures and organic solvents are exploited in biotechnology.
Classification. New domain.
For much of the 20th century, prokaryotes were regarded as a single group of organisms and classified based on their biochemistry, morphology and metabolism. For example, microbiologists tried to classify microorganisms based on the structures of their cell walls, their shapes, and the substances they consume. However, a new approach was proposed in 1965, using the sequences of the genes in these organisms to work out how different prokaryotes are related to each other. This approach, known as phylogenetics, is the main method used today. Archaea were first classified as a separate group of prokaryotes in 1977 by Carl Owies and George E. Fox in phylogenetic trees based on the sequences of ribosomal RNA, or RNA, genes. These two groups were originally named the Archaebacteria and Eubacteria and treated as kingdoms or subkingdoms, which Owies and Fox termed Urkingdoms. Owies argued that this group of prokaryotes is a fundamentally different sort of life. To emphasize this difference, these two domains were later renamed Archaean bacteria. The word Archaea comes from the ancient Greek archaea, meaning ancient things. The first representatives of the domain Archaea were methangens. For a long time Archaea were seen as extremophiles that only exist in extreme habitats such as hot springs and salt lakes. However, by the end of the 20th century, Archaea had been identified in non-extreme environments as well. Today they are known to be a large and diverse group of organisms that are widely distributed in nature and are common in all habitats. This new appreciation of the importance and ubiquity of Archaea came from using polymerase chain reaction, PCR, to detect prokaryotes from environmental samples, such as water or soil, by multiplying their ribosomal genes. This allows the detection and identification of organisms that have not been cultured in the laboratory.
Current classification.
The classification of Archaea, and of prokaryotes in general, is a rapidly moving and contentious field. Current classification systems aim to organize Archaea into groups of organisms that share structural features and common ancestors. These classifications rely heavily on the use of the sequence of ribosomal RNA genes to reveal relationships between organisms, molecular phylogenetics. Most of the culturable and well-investigated species of Archaea are members of two main phyla, the Archaea and Archaea. Other groups have been tentatively created. For example, the peculiar species Archaea mequitans, which was discovered in 2003, has been given its own phylum, the Archaea. A new phylum Archaea has also been proposed. It contains a small group of unusual thermophilic species that shares features of both of the main phyla, but is most closely related to the Archaea. Other recently detected species of Archaea only distantly related to any of these groups, such as the archaeal Richmond-Mine acidophilic nanoorganisms, ARMAN, which were discovered in 2006 and are some of the smallest organisms known. A superphylum, TAC, has been proposed that includes the Archaea, Archaea, Archaea and Archaea. This superphylum may be related to the origin of eukaryotes.
Species.
The classification of Archaea into species is also controversial. Biology defines a species as a group of related organisms. The familiar exclusive breeding criterion, organisms that can breed with each other but not with others, is of no help because Archaea reproduce asexually. Archaea show high levels of horizontal gene transfer between lineages. Some researchers suggest that individuals can be grouped into species-like populations given highly similar genomes and infrequent gene transfer from cells with less related genomes, as in the genus Ferroplasma. On the other hand, studies in Holoubrum found significant genetic transfer from less related populations, limiting the criterion's applicability. A second concern is to what extent such species designations have practical meaning. Current knowledge on genetic diversity is fragmentary and the total number of archaeal species cannot be estimated with any accuracy. Estimates of the number of phyla range from 18 to 23, of which only 8 have representatives that have been cultured and studied directly. Many of these hypothesized groups are known from a single RNA sequence, indicating that the diversity among these organisms remains obscure. The bacteria also contain many uncultured microbes with similar implications for characterization.
Origin and evolution.
Scientific evidence suggests that life began on Earth at least 3.5 billion years ago. The earliest evidences for life on Earth are graphite found to be biogenic in 3.7 billion-year-old metasedimentary rocks discovered in western Greenland and microbial mat fossils found in 3.48 billion-year-old sandstone discovered in Western Australia. Although probable prokaryotic cell fossils date to almost 3.5 billion years ago, most prokaryotes do not have distinctive morphologies and fossil shapes cannot be used to identify them as Archaea. Instead, chemical fossils of unique lipids are more informative because such compounds do not occur in other organisms. Some publications suggest that archaeal or eukaryotic lipid remains are present in shales dating from 2.7 billion years ago. Such data have since been questioned. Such lipids have also been detected in even older rocks from West Greenland. The oldest such traces come from the Yasua district, which include Earth's oldest sediments, formed 3.8 billion years ago. The Archaea lineage may be the most ancient that exists on Earth. Owies argued that the bacteria, Archaea, and eukaryotes represent separate lines of descent that diverged early on from an ancestral colony of organisms. One possibility is that this occurred before the evolution of cells, when the lack of a typical cell membrane allowed unrestricted lateral gene transfer, and that the common ancestors of the three domains arose by fixation of specific subsets of genes. It is possible that the last common ancestor of the bacteria and Archaea was a thermophile, which raises the possibility that lower temperatures are extreme environments in archaeal terms, and organisms that live in cooler environments appeared only later. Since the Archaean bacteria are no more related to each other than they are to eukaryotes, the term prokaryotes only surviving meaning is not a eukaryote, limiting its value.
Comparison to other domains.
The following table describes some major characteristics that are generally shared by Archaea with the other two domains or with neither, to illustrate the relationships to each. Many of these characteristics are also discussed below.
Relationship to other prokaryotes.
The relationship between the three domains is of central importance for understanding the origin of life. Most of the metabolic pathways, which comprise the majority of an organism's genes, are common between Archaea and bacteria, while most genes involved in genome expression are common between Archaea and Eukarya. Within prokaryotes, archaeal cell structure is most similar to that of gram-positive bacteria, largely because both have a single lipid bilayer and usually contain a thick circulus of varying chemical composition. In phylogenetic trees based upon different gene-protein sequences of prokaryotic homologs, the archaeal homologs are more closely related to those of gram-positive bacteria. Archaea and gram-positive bacteria also share conserved indels in a number of important proteins, such as HSP70 and glutamine synthetase I. R.S. Gupta has proposed that the Archaea evolved from gram-positive bacteria in response to antibiotic selection pressure. This is suggested by the observation that Archaea are resistant to a wide variety of antibiotics that are primarily produced by gram-positive bacteria, and that these antibiotics primarily act on the genes that distinguish Archaea from bacteria. His proposal is that the selective pressure towards resistance generated by the gram-positive antibiotics was eventually sufficient to cause extensive changes in many of the antibiotics' target genes, and that these strains represented the common ancestors of present-day Archaea. The evolution of Archaea in response to antibiotic selection, or any other competitive selective pressure, could also explain their adaptation to extreme environments, such as high temperature or acidity, as the result of a search for unoccupied niches to escape from antibiotic-producing organisms. Cavalier-Smith has made a similar suggestion. Gupta's proposal is also supported by other work investigating protein structural relationships and studies that suggest that gram-positive bacteria may constitute the earliest branching lineages within the prokaryotes.
Relation to eukaryotes.
The evolutionary relationship between Archaea and eukaryotes remains unclear. Aside from the similarities in cell structure and function that are discussed below, many genetic trees group the two. Complicating factors include claims that the relationship between eukaryotes and the archaeal phylum Cranarchaeota is closer than the relationship between the Euarchaeota and the phylum Cranarchaeota, and the presence of Archaea-like genes in certain bacteria, such as Thermotoca maritima, from horizontal gene transfer. The standard hypothesis states that the ancestor of the eukaryotes diverged early from the Archaea, and that eukaryotes arose through fusion of an archaean and eubacterium, which became the nucleus and cytoplasm. This explains various genetic similarities but runs into difficulties explaining cell structure. An alternative hypothesis, the Eocyte hypothesis, posits that Eukaryota emerged relatively late from the Archaea.
Morphology.
Individual Archaea range from 0.1 micrometers, to over 15 millimeters in diameter, and occur in various shapes, commonly as spheres, rods, spirals or plates. Other morphologies in the Cranarchaeota include irregularly shaped lobe cells in sulfolobus, needle-like filaments that are less than half a micrometer in diameter in Thermophilum, and almost perfectly rectangular rods in Thermoproteus and Pyrobaculum. Haloquadratum Wallsby are flat, square Archaea that live in hypersaline pools. These unusual shapes are probably maintained both by their cell walls and a prokaryotic cytoskeleton. Proteins related to the cytoskeleton components of other organisms exist in Archaea, and filaments form within their cells, but in contrast to other organisms, these cellular structures are poorly understood. In thermoplasma and ferroplasma, the lack of a cell wall means that the cells have irregular shapes, and can resemble amoebae. Some species form aggregates or filaments of cells up to 200 mm long. These organisms can be prominent in biofilms. Notably, aggregates of Thermococcus coalescens cells fuse together in culture, forming single giant cells. Archaea in the genus Pyrodictium produce an elaborate multi-cell colony involving arrays of long, thin hollow tubes called cannulae that stick out from the cell's surfaces and connect them into a dense bush-like agglomeration. The function of these cannulae is not settled, but they may allow communication or nutrient exchange with neighbors. Multi-species colonies exist, such as the String of Pearls community that was discovered in 2001 in a German swamp. Round whitish colonies of a novel Archaea species are spaced along thin filaments that can range up to 15 cm, 5.9 inches, long. These filaments are made of a particular bacteria species.
Structure, composition, development, operation.
Archaean bacteria have generally similar cell structure, but cell composition and organization set the Archaea apart. Like bacteria, Archaea lack interior membranes and organelles. Like bacteria, Archaea cell membranes are usually bounded by a cell wall and they swim using one or more flagella. Structurally, Archaea are most similar to gram-positive bacteria. Most have a single plasma membrane and cell wall, and lack a periplasmic space. The exception to this general rule is Ignicoccus, which possess a particularly large periplasm that contains membrane-bound vesicles and is enclosed by an outer membrane.
Membranes.
Archaeal membranes are made of molecules that differ strongly from those in other life forms, showing that Archaea are related only distantly to bacteria and eukaryotes. In all organisms, cell membranes are made of molecules known as phospholipids. These molecules possess both a polar part that dissolves in water, the phosphate head, and a greasy non-polar part that does not, the lipid tail. These dissimilar parts are connected by a glycerol moiety. In water, phospholipids cluster, with the heads facing the water and the tails facing away from it. The major structure in cell membranes is a double layer of these phospholipids, which is called a lipid bilayer. These phospholipids are unusual in four ways. Bacteria and eukaryotes have membranes composed mainly of glycerol ester lipids, whereas Archaea have membranes composed of glycerol ether lipids. The difference is the type of bond that joins the lipids to the glycerol moiety. The two types are shown in yellow in the figure at the right. In ester lipids this is an ester bond, whereas in ether lipids this is an ether bond. Ether bonds are chemically more resistant than ester bonds. This stability might help Archaea to survive extreme temperatures and very acidic or alkaline environments. Bacteria and eukaryotes do contain some ether lipids, but in contrast to Archaea, these lipids are not a major part of their membranes. The stereochemistry of the glycerol moiety is the reverse of that found in other organisms. The glycerol moiety can occur in two forms that are mirror images of one another, called the right-handed and left-handed forms. In chemistry these are called enantiomers. Just as a right hand does not fit easily into a left-handed glove, a right-handed phospholipid generally cannot be used or made by enzymes adapted for the left-handed form. This suggests that Archaea use entirely different enzymes for synthesizing phospholipids than do bacteria and eukaryotes. Such enzymes developed very early in life's history, suggesting an early split from the other two domains. Archaea lipid tails are chemically different from other organisms. Archaea lipids are based upon the isoprenoid side chain and are long chains with multiple side branches and sometimes even cyclopropane or cyclohexane rings. This is in contrast to the fatty acids found in other organisms' membranes, which have straight chains with no branches or rings. Although isoprenoids play an important role in the biochemistry of many organisms, only the Archaea use them to make phospholipids. These branched chains may help prevent archaeal membranes from leaking at high temperatures. In some Archaea the lipid bilayer is replaced by a monolayer. In effect, the Archaea fuse the tails of two independent phospholipid molecules into a single molecule with two polar heads, a ballonophile. This fusion may make their membranes more rigid and better able to resist harsh environments. For example, the lipids in ferroplasma are of this type, which is thought to aid this organism's survival in its highly acidic habitat.
Wall and flagella.
Most Archaea, but not thermoplasma and ferroplasma, possess a cell wall. In most Archaea the wall is assembled from surface layer proteins, which form an S-layer. An S-layer is a rigid array of protein molecules that cover the outside of the cell, like chain mail. This layer provides both chemical and physical protection, and can prevent macromolecules from contacting the cell membrane. Unlike bacteria, Archaea lack peptidoglycan in their cell walls. Methanobacteriales do have cell walls containing pseudopeptidoglycan, which resembles eubacterial peptidoglycan in morphology, function, and physical structure, but pseudopeptidoglycan is distinct in chemical structure. It lacks D-amino acids and N-acetylmuramic acid. Archaea flagella operate like bacterial flagella. Their long stalks are driven by rotatory motors at the base. These motors are powered by the proton gradient across the membrane. However, archaeal flagella are notably different in composition and development. The two types of flagella evolved from different ancestors. The bacterial flagellum shares a common ancestor with the type 3 secretion system, while archaeal flagella appear to have evolved from bacterial type 4 pili. In contrast to the bacterial flagellum, which is hollow and is assembled by subunits moving up the central pore to the tip of the flagella, archaeal flagella are synthesized by adding subunits at the base.
Metabolism.
Archaea exhibit a great variety of chemical reactions in their metabolism and use many sources of energy. These reactions are classified into nutritional groups, depending on energy and carbon sources. Some Archaea obtain energy from inorganic compounds such as sulfur or ammonia. They are lithotrophs. These include nitrifiers, methangens and anaerobic methane oxidizers. In these reactions one compound passes electrons to another, in a redox reaction, releasing energy to fuel the cell's activities. One compound acts as an electron donor and one as an electron acceptor. The energy released generates adenosine triphosphate, ATP, through chemiosmosis, in the same basic process that happens in the mitochondrion of eukaryotic cells. Other groups of Archaea use sunlight as a source of energy. They are phototrophs. However, oxygen-generating photosynthesis does not occur in any of these organisms. Many basic metabolic pathways are shared between all forms of life. For example, Archaea use a modified form of glycolysis, the Entner-Doudoroff pathway, and either a complete or partial citric acid cycle. These similarities to other organisms probably reflect both early origins in the history of life and their high level of efficiency. Some Eukaryotes are methangens living in anaerobic environments such as swamps. This form of metabolism evolved early, and it is even possible that the first free-living organism was a methangen. A common reaction involves the use of carbon dioxide as an electron acceptor to oxidize hydrogen. Methanogenesis involves a range of coenzymes that are unique to these Archaea, such as coenzyme M and methanofuran. Other organic compounds such as alcohols, acetic acid or formic acid are used as alternative electron acceptors by methangens. These reactions are common in gut-dwelling Archaea. Acetic acid is also broken down into methane and carbon dioxide directly, by acetotrophic Archaea. These acetotrophs are Archaea in the order Methanosarcinales, and are a major part of the communities of microorganisms that produce biogas. Other Archaea use CO2 in the atmosphere as a source of carbon. In a process called carbon fixation, they are autotrophs. This process involves either a highly modified form of the Calvin cycle or a recently discovered metabolic pathway called the 3-hydroxypropionate/4-hydroxybutyrate cycle. The Cranarchaea also use the reverse Krebs cycle while the Eukaryotes also use the reductive acetyl-CoA pathway. Carbon fixation is powered by inorganic energy sources. No known Archaea carry out photosyntheses. Archaeal energy sources are extremely diverse. and range from the oxidation of ammonia by the nitrosapumaleals to the oxidation of hydrogen sulfide or elemental sulfur by species of sulfolobus, using either oxygen or metal ions as electron acceptors. Phototrophic Archaea use light to produce chemical energy in the form of ATP. In the halobacteria, light-activated ion pumps like bacteriorhodopsin and halorhodopsin generate ion gradients by pumping ions out of the cell across the plasma membrane. The energy stored in these electrochemical gradients is then converted into ATP by ATP synthase. This process is a form of photophosphorylation. The ability of these light-driven pumps to move ions across membranes depends on light-driven changes in the structure of a retinol cofactor buried in the center of the protein.
Genetics.
Archaea usually have a single circular chromosome, the size of which may be as great as 5,751,492 base pairs in Methanosarcina astivirans, the largest known archaeal genome. 1/10 of this size is the tiny 490,885 base paired genome of Archaea mequitans, the smallest archaeal genome known. It is estimated to contain only 537 protein-encoding genes. Smaller independent pieces of DNA, called plasmids, are also found in Archaea. Plasmids may be transferred between cells by physical contact, in a process that may be similar to bacterial conjugation. Archaea can be infected by double-stranded DNA viruses that are unrelated to any other form of virus and have a variety of unusual shapes, including bottles, hooked rods, or teardrops. These viruses have been studied in most detail in thermophilics, particularly the orders sulfolobales and amoproteales. Two groups of single-stranded DNA viruses that infect Archaea have been recently isolated. One group is exemplified by the haloeubrim pleomorphic virus 1, Plolipoviridae infecting halophilic Archaea, and the other one by the aeropyrum coil-shaped virus, Sporaviridae infecting a hyperthermophilic, optimal growth at 90 to 95 DEGC, host. Notably, the latter virus has the largest currently reported ssDNA genome. Defenses against these viruses may involve RNA interference from repetitive DNA sequences that are related to the genes of the viruses. Archaea are genetically distinct from bacteria and eukaryotes, with up to 15% of the proteins encoded by any one archaeal genome being unique to the domain, although most of these unique genes have no known function. Of the remainder of the unique proteins that have an identified function, most belong to the Archaea and are involved in methanogenesis. The proteins that Archaea, bacteria and eukaryotes share form a common core of cell function, relating mostly to transcription, translation, and nucleotide metabolism. Other characteristic archaeal features are the organization of genes of related function, such as enzymes that catalyze steps in the same metabolic pathway into novel operons, and large differences in tRNA genes and their aminosyl tRNA synthetases. Transcription and translation in Archaea resemble these processes in eukaryotes more than in bacteria, with the archaeal RNA polymerase and ribosomes being very close to their equivalents in eukaryotes. Although Archaea only have one type of RNA polymerase, its structure and function in transcription seems to be close to that of the eukaryotic RNA polymerase II, with similar protein assemblies, the general transcription factors, directing the binding of the RNA polymerase to a gene's promoter. However, other archaeal transcription factors are closer to those found in bacteria. Post-transcriptional modification is simpler than in eukaryotes, since most archaeal genes lack introns, although there are many introns in the transfer RNA and ribosomal RNA genes, and introns may occur in a few protein-encoding genes.
Gene transfer and genetic exchange.
Halobacterium volcania, an extreme halophilic archaeon, forms cytoplasmic bridges between cells that appear to be used for transfer of DNA from one cell to another in either direction. When the hyperthermophilic Archaea sulfolobus sulfataricus and sulfolobus acidocolderius are exposed to the DNA-damaging agents UV irradiation, bleomycin or mutamycin C, species-specific cellular aggregation is induced. Aggregation in S-sulfur Taricus could not be induced by other physical stressors, such as pH or temperature shift, suggesting that aggregation is induced specifically by DNA damage. Archaea showed that UV-induced cellular aggregation mediates chromosomal marker exchange with high frequency in S. acidocolderias. Recombination rates exceeded those of uninduced cultures by up to 3 orders of magnitude. Frols et al. And Archaea hypothesized that cellular aggregation enhances species-specific DNA transfer between sulfur-lobose cells in order to provide increased repair of damaged DNA by means of homologous recombination. This response may be a primitive form of sexual interaction similar to the more well-studied bacterial transformation systems that are also associated with species-specific DNA transfer between cells leading to homologous recombinational repair of DNA damage.
Reproduction.
Archaea reproduce asexually by binary or multiple fission, fragmentation, or budding. Meiosis does not occur, so if a species of Archaea exists in more than one form, all have the same genetic material. Cell division is controlled in a cell cycle. After the cell's chromosome is replicated and the 2 daughter chromosomes separate, the cell divides. Details have only been investigated in the genus Sulfur-lobose, but that cycle has characteristics that are similar to both bacterial and eukaryotic systems. The chromosomes replicate from multiple starting points, origins of replication, using DNA polymerases that resemble the equivalent eukaryotic enzymes. However, the proteins that direct cell division, such as the protein FTSZ, which forms a contracting ring around the cell, and the components of the septum that is constructed across the center of the cell, are similar to their bacterial equivalents. Both bacteria and eukaryotes, but not Archaea, make spores. Some species of Archaea undergo phenotypic switching and grow as several different cell types, including thick-walled structures that are resistant to osmotic shock and allow the Archaea to survive in water at low salt concentrations, but these are not reproductive structures and may instead help them reach new habitats.
Ecology. Habitats.
Archaea exist in a broad range of habitats, and as a major part of global ecosystems, may contribute up to 20% of Earth's biomass. The first discovered archaeans were extremophiles. Indeed, some Archaea survive high temperatures, often above 100 DEGC, 212 DEGF, as found in geysers, black smokers, and oil wells. Other common habitats include very cold habitats and highly saline, acidic, or alkaline water. However, Archaea include mesophiles that grow in mild conditions, in marshland, sewage, the oceans, and soils. Extremophile Archaea are members of 4 main physiological groups. These are the holophiles, thermophiles, alkalophiles, and acidophiles. These groups are not comprehensive or phylum-specific, nor are they mutually exclusive, since some Archaea belong to several groups. Nonetheless, they are a useful starting point for classification. Holophiles, including the genus Halobacterium, live in extremely saline environments such as salt lakes and outnumber their bacterial counterparts at salinities greater than 20 to 25%. Thermophiles grow best at temperatures above 45 DEGC, 113 DEGF, in places such as hot springs. Hyperthermophilic Archaea grow optimally at temperatures greater than 80 DEGC, 176 DEGF. The Archaeal Methanopyrus candelari strain 116 can even reproduce at 122 DEGC, 252 DEGF, the highest recorded temperature of any organism. Other Archaea exist in very acidic or alkaline conditions. For example, one of the most extreme Archaean acidophiles is Picrophilus torridus, which grows at penthouse O, which is equivalent to thriving in 1.2 molar sulfuric acid. This resistance to extreme environments has made Archaea the focus of speculation about the possible properties of extraterrestrial life. Some extremophile habitats are not dissimilar to those on Mars, leading to the suggestion that viable microbes could be transferred between planets in meteorites. Recently, Several studies have shown that Archaea exist not only in mesophilic and thermophilic environments but are also present, sometimes in high numbers, at low temperatures as well. For example, Archaea are common in cold oceanic environments such as polar seas. Even more significant are the large numbers of Archaea found throughout the world's oceans in non-extreme habitats among the plankton community, as part of the phytoplankton. Although these Archaea can be present in extremely high numbers, up to 40% of the microbial biomass, almost none of these species have been isolated and studied in pure culture. Consequently, our understanding of the role of Archaea in ocean ecology is rudimentary, so their full influence on global biogeochemical cycles remains largely unexplored. Some marine Archaea are capable of nitrification, suggesting these organisms may affect the oceanic nitrogen cycle, although these oceanic Archaea may also use other sources of energy. Vast numbers of Archaea are also found in the sediments that cover the sea floor, with these organisms making up the majority of living cells at depths over 1 meter below the ocean bottom.
Role in chemical cycling.
Archaea recycle elements such as carbon, nitrogen and sulfur through their various habitats. Although these activities are vital for normal ecosystem function, Archaea can also contribute to human-made changes, and even cause pollution. Archaea carry out many steps in the nitrogen cycle. This includes both reactions that remove nitrogen from ecosystems, such as nitrate-based respiration and denitrification, as well as processes that introduce nitrogen, such as nitrate assimilation and nitrogen fixation. Archaean involvement in ammonia oxidation reactions was recently discovered. These reactions are particularly important in the oceans. The Archaea also appear to be crucial for ammonia oxidation in soils. They produce nitrite, which other microbes then oxidize to nitrate. Plants and other organisms consume the latter. In the sulfur cycle, Archaea that grow by oxidizing sulfur compounds release this element from rocks, making it available to other organisms. However, the Archaea that do this, such as sulfur locus, produce sulfuric acid as a waste product, and the growth of these organisms in abandoned mines can contribute to acid mine drainage and other environmental damage. In the carbon cycle, methangen Archaea remove hydrogen and are important in the decay of organic matter by the populations of microorganisms that act as decomposers in anaerobic ecosystems, such as sediments, marshes and sewage treatment works. Methangens are the primary source of atmospheric methane, and are responsible for most of the world's yearly methane emissions. As a consequence, these Archaea contribute to global greenhouse gas emissions and global warming. Global methane levels have risen to 1800 ppb by 2011, an increase by a factor of 2.5 since pre-industrial times, from 722 ppb, the highest value in at least 800,000 years. Methane has an anthropogenic global warming potential, AGWP, of 29, which means that it's 29 times stronger in heat trapping than carbon dioxide is, over a 100-year time scale.
Interactions with other organisms.
The well-characterized interactions between Archaea and other organisms are either mutual or commensal. There are no clear examples of known archaeal pathogens or parasites. However, a relationship has been proposed between some species of methangens and infections in the mouth, and Archaea mequitans may be a parasite of another species of Archaea, since it only survives and reproduces within the cells of the Archaea and Ignicoccus hospitalis, and appears to offer no benefit to its host. In contrast, archaeal Richmond-mine acidophilic nanoorganisms, ARMAN, occasionally connect with other archaeal cells in acid mine drainage biofilms. The nature of this relationship is unknown. However, it is distinct from that of Archaea Ignicoccus in that the ultra-small ARMAN cells are usually seen independent of the thermoplasma tail cells.
Mutualism.
One well-understood example of mutualism is the interaction between protozoa and methanogenic Archaea in the digestive tracts of animals that digest cellulose, such as ruminants and termites. In these anaerobic environments, protozoa break down plant cellulose to obtain energy. This process releases hydrogen as a waste product, but high levels of hydrogen reduce energy production. When methangens convert hydrogen to methane, protozoa benefit from more energy. In anaerobic protozoa such as Plagiopula frontata, Archaea reside inside the protozoa and consume hydrogen produced in their hydrogenosomes. Archaea also associate with larger organisms. For example, the marine Archaean Synachaeum symbiosum lives within, is an endosymbiont of, the sponge Xinella mexiusana.
Commensalism.
Archaea can also be commensals, benefiting from an association without helping or harming the other organism. For example, the methangen Methanobravibacter smithii is by far the most common archaean in the human flora, making up about 1 in 10 of all the prokaryotes in the human gut. In termites and in humans, these methangens may in fact be mutualists, interacting with other microbes in the gut to aid digestion. Archaean communities also associate with a range of other organisms, such as on the surface of corals, and in the region of soil that surrounds plant roots, the rhizosphere.
Significance in technology and industry.
Extremophile Archaea, particularly those resistant either to heat or to extremes of acidity and alkalinity, are a source of enzymes that function under these harsh conditions. These enzymes have found many uses. For example, the mostable DNA polymerases, such as the PEW DNA polymerase from Pyrococcus furiosus, revolutionized molecular biology by allowing the polymerase chain reaction to be used in research as a simple and rapid technique for cloning DNA. In industry, amylases, galactosidases and polyolinases and other species of Pyrococcus that function at over 100 DEGC, 212 DEGF, allow food processing at high temperatures, such as the production of low-lactose milk and whey. Enzymes from these thermophilic Archaea also tend to be very stable in organic solvents, allowing their use in environmentally friendly processes in green chemistry that synthesize organic compounds. This stability makes them easier to use in structural biology. Consequently, the counterparts of bacterial or eukaryotic enzymes from extremophile Archaea are often used in structural studies. In contrast to the range of applications of archaean enzymes, the use of the organisms themselves in biotechnology is less developed. Methanogenic Archaea are a vital part of sewage treatment, since they are part of the community of microorganisms that carry out anaerobic digestion and produce biogas. In mineral processing, acidophilic Archaea display promise for the extraction of metals from ores, including gold, cobalt and copper. Archaea host a new class of potentially useful antibiotics. A few of these archaeocins have been characterized, but hundreds more are believed to exist, especially within Archaean sulfur lobbers. These compounds differ in structure from bacterial antibiotics, so they may have novel modes of action. In addition, they may allow the creation of new selectable markers for use in archaeal molecular biology.